Every single second, inside trillions of your cells, something absolutely mind-boggling is happening. It's this incredibly precise, complex process that's the very foundation of growth, of repair, of life itself. So let's dive into this masterclass of molecular engineering, DNA replication.
And honestly, calling it a trick might be an understatement. We are talking about a process that has to copy the entire instruction manual for a living being, and it has to do it perfectly, time after time. So just how big is that instruction manual we're talking about?
3 billion. That's the number of letters, or what we call base pairs, in your genome. To put that in perspective, that's like copying a huge stack of books, thousands of them, letter for letter, without making a mistake.
And your cells? They pull off this incredible feat every single time they divide. They have to make sure each new daughter cell gets its own complete, perfect copy of the blueprint. This, right here, is how life makes more life. So how on earth does it work? What are the rules of the game?
Well, the secret to all this amazing accuracy, it lies in a beautifully simple principle. The DNA molecule itself actually holds the key to its own duplication.
The whole thing is called semi-conservative replication. It sounds complicated, but it's not. Semi just means half, and conservative means saved. So each new DNA molecule saves half of the original. This one idea is the key to the whole thing.
Okay, let's walk through it real quick. You start with one parent DNA molecule, right? Then the two strands unzip, kind of like a zipper. Now, each of those original strands becomes a perfect template for a new one. The cell's machinery just reads that template and builds a brand new partner strand right alongside it. And the result? You get two identical DNA molecules, and each one is a perfect hybrid, half old, half new. But of course, this process doesn't just happen. It needs a whole team of molecular machines to get the job done.
So let's meet the major players on this replication team. And a good way to think about it is as a series of problems that the cell has to solve. All right, first problem. The DNA double helix is really stable. I mean, those two strands are wound together super tightly. You can't copy something if you can't read it. So how do you get inside?
Well, you bring in an enzyme called helicase. Just think of it as a molecular unzipper. Its whole job is to move along the DNA, breaking the bonds between the base pairs and prying those two strands apart. This creates this Y-shaped area called the replication fork, and that's where all the action's going to happen.
Okay, so the strands are open, they're exposed. We have our templates ready to go. Now for the main event. How do we actually build the new strands of DNA?
This is the job for the star of the show, an enzyme called DNA polymerase. This thing is the master builder. It zips along the template strand, reads the sequence of letters, and then grabs the matching nucleotide from the cellular soup, A for T, G for C, and links them all together to form the new strand.
But of course, there's a catch. There's always a catch, right? DNA polymerase is particular. It has a strict one-way street rule. See, the two DNA strands run in opposite directions. Think of it like a highway with two lanes of traffic, going opposite ways. Well, DNA polymerase can only drive in one of those directions. This creates a really interesting problem for the cell, which, needs to copy both lanes at the same time. So because of this one-way rule, the two new strands get built in totally different ways. One strand, we call it the leading strand, is super easy. It's pointed in the perfect direction for DNA polymerase to just cruise along, building one long, continuous piece as the fork unzips. But the other strand, the lagging strand, that's the tricky one. It's pointed the wrong way. So to copy it, the polymerase has to work backwards, building it in a series of short, little disconnected chunks.
And these short little segments have a name. They're called Okazaki fragments, named after the scientists who discovered them. It's pretty clever, really. The cell basically makes a little piece, then it hops back towards the fork where more of the strand is opened up, and it makes another little piece. It's kind of like a backstitching motion.
So this whole backstitching thing solves the direction problem, but it creates a new one. Now the lagging strand is just a bunch of disconnected fragments, and that's not going to work. So how does the cell patch it all up and make one seamless strand? This is where our final player comes in, DNA ligase. Its job is simple, but absolutely critical. It acts like molecular glue. It goes along the lagging strand and seals up all the gaps between those Okazaki fragments, creating one continuous, complete strand of DNA. Job done.
So we have this amazing molecular machine that can copy 3 billion letters.
But just being fast isn't enough, right? It has to be perfect. Even one tiny mistake in the genetic code can be a disaster. So how does the cell make sure it gets it right?
Well, this process doesn't just rely on luck. The whole system has built-in quality control. We're talking about multiple layers of proofreading and error correction that catch and fix mistakes almost as soon as they're made. Just how accurate are we talking?
The final error rate after all the checks and balances are done is about 1 mistake for every billion letters copied. That's insane. That is like making a single typo while copying over 1,000 full-length novels.
And here's how it gets there. See, just the basic rules of base pairing get you to about 1 mistake in 100,000. That's pretty good, but not great. But then, DNA polymerase itself does its own proofreading. Right after it adds a new letter, it kind of double-checks its work. If it's wrong, it snips it out and tries again. That simple step improves accuracy 100 times, down to 1 in 10 million. And finally, there's another system called mismatch repair that scans the new DNA after it's made, catching any mistakes the polymerase might have missed. That final check is what gets us to that unbelievable 1 in a billion number.
But what about that 1 in a billion? It's a tiny chance, but it's not zero. What happens when a mistake does manage to slip through all of those defenses?
When an error isn't caught, it gets locked into the DNA sequence for good. And from that point on, it'll be copied over and over in every future cell division. That permanent change has a name. It's called a mutation.
Now, a lot of mutations don't really do anything, but some can be incredibly harmful. You can really see how important this proofreading is when the system breaks. For example, people who inherit a broken mismatch repair gene have a much higher risk for certain cancers, like hereditary non-polyposis colon cancer. Why? Because their cells are just accumulating mistakes at a much faster rate. It really shows you that this quest for perfection is a matter of life and death.
From the simple elegance of using an old strand to make a new one, to this incredibly complex dance of enzymes, DNA replication is the process that connects every single generation of life on this planet. It's a system of truly breathtaking precision, but it's not perfect. And it's in that tiny 1 in a billion chance of an error where we find the origins of genetic disease, but also the engine of evolution and the very source of all the diversity of life itself.